Hello everyone, today we are talking about the difference between bacteria and virus. Bacteria is a prokaryotic organism and is nearly involved in everything, like food production, human body, and also industry. And now we will talk about bacteria structure. Bacteria lack a true nucleus and membrane-bound organelles. First is the cell wall. Cell wall provides shape and also protection. It is made of peptidoglycan in most bacteria. Then we have the plasma membrane. It is a phospholipid bilayer that regulates the movement of substance in and out of the cell. Number 3 is the capsule. It is a slimy outer layer that protects against dissection and also immune responses. Number 4 is the flagella. It is a tail-like structure that enable movement. Then number 5 is the pili. It is a hair-like structure that helps in the attachment to surface and also genetic exchange. Number 6 is the cytoplasm. It contains enzymes, nutrients, and ribosomes. Then number 7 is the nucleoid. It is a region containing the bacterial DNA. Next we have the plasmids. It is small circular DNA molecules that carry extra genes such as antibiotic resistant genes. And lastly is the ribosomes, and they are responsible for protein synthesis. Now we will talk about bacteria size and shapes. Bacteria are microscopic single-celled organisms that range from 0.2 to 5 micrometers in length. And we have 3 shapes for bacteria. The first one is the cocci. It is round or oval-shaped bacteria. Number 2 is the bacilli. It is cylindrical or elongated bacteria. And the last shape is the Spirochetes. It is flexible helical bacteria with a corkscrew motion. And now we will talk about bacteria common diseases. The first one is the respiratory diseases, like pneumonia, like the Streptococcus pneumoniae, and it leads to fever, cough with mucus, chest pain, and also difficulty breathing. And number 2 is a skin disease. For example, impetigo, like Staphylococcus aureus, and it is a contiguous skin infection with red sores and yellow crust. And number 3 is the digestive tract disease, like food poisoning. It is caused by salmonella, and it causes nausea, vomiting, diarrhea, and stomach cramps. And now we will talk about bacteria ways of infection. Number 1 is a direct contact. Bacteria spread through physical contact with an infected person, wind, or contaminated surface. Number 2 is the contaminated food and water. Bacteria enter the body through ingestion of contaminated food or water. For example, Salmonella and also E. coli. Number 3 is the vertical transmission. It is bacteria that can be passed from mother to baby during pregnancy, childbirth, or breastfeeding, like the group B Streptococcus. And now we will talk about virus. Virus is a non-living strand of genetic material within a protein coat, and it is believed that it is non-living because it lacks most organelles. Also, they cannot move on their own, and also they cannot replicate on their own. And now let's talk about virus structure. Number 1 is the capsid, and it is a protein coat that encases and also protects the virus genetic material. Also, it is made up of protein subunits called capsomers, and also it gives the virus its shape. Number 2 is the genetic material. Virus contain either DNA or RNA, but not both. And it carries the genetic an instruction for virus replication inside the host. Number 3 is the tail. It is a tube-like structure used by some viruses, like bacteriophages, to inject genetic material into host cells. Number 4 is the tail fibers. It is a hair-like projections extending from the tail. Also, it helps in the attaching to a host cells by recognizing specific receptors. Number 5 is the envelope. It is a lipid membrane derived from the host cell, and it surrounds the capsid and also provides extra protection. And it is found in viruses like influenza and also HIV. Number 6 is the spikes. It is a protein projections on the envelope or capsid, and it helps in the attachment to host cells and entry. Example, like the spike proteins of the coronavirus. And lastly, number 7 is the protein units. It is a structured protein that make up the capsid and also envelope, and it helps in viral assembly and stability. And now let's talk about virus size. Viruses are some of the smallest disease-causing structures. Most of them, they range from 5 to 300 nanometers. And now let's talk about virus shapes. The first one is the adenovirus, and it is an icosahedral shape. The capsid has a 20-sided symmetry. And the example is like it causes respiratory infections. Second type is the influenza virus, and it is spherical shaped. It is involved with a roughly spherical shape, but can be slightly variable. Also, it contains the helical nucleocapsid inside. Example, like it causes the flu. Number 3 is the bacteriophage, and it has a complex shape. The head is the icosahedral capsid containing DNA, and the tail hollow tube for injecting DNA into bacteria, and the tail fibers help in the attachment. Example, T4 phage infects E. coli. And the fourth type is the tobacco mosaic virus. It is a rigid rod-like structure, helical capsid that surrounds RNA. Example, it infects plants like tobacco and tomatoes. And now let's talk about common diseases caused by virus. First is the skin-like shingles. Shingles is a viral infection caused by the same virus that causes chickenpox. And it happened because after a person that recovers from the chickenpox, the virus remains dormant in the nerve tissues. It can reactive later in life as shingles. Next, we have the sexually transmitted disease, like AIDS. It is a sexually transmitted disease caused by the human immune defense virus. Next, we have childhood diseases, like chickenpox. It is a highly contagious viral infection caused by the varicella zoster virus, and it is primarily affects children, but it can occur in the adults who haven't been vaccinated or previously infected. Lastly, we have the respiratory diseases, like common colds. It is caused by rhinovirus, leading to a runny nose and cough. Also, we have pneumonia. It is lung infection caused by bacteria, viruses, or fungi. This cycle process is when the virus attacks the host cell machinery to produce new viruses, eventually destroying the host cell to release them. And the first step is the attachment. The virus attaches to a specific receptor on the surface of the host cell. Second is the entry, is when the viral genetic material DNA or RNA enters the host cell. Third is the replication, is when the virus takes over the host cell machinery to replicate its genetic material and proteins. Number 4 is the assembly. New viral particles are now assembled from the copied genetic material and proteins. Then the last step is the release. It is when the host cell bursts, releasing hundreds of new viruses. And the next cycle is the lysogenic cycle. It is when the viral DNA inserts or integrates into a chromosome in the host cell. And the first step is the attachment. It is when the virus binds to a specific receptor on the host cell surface. Number 2 is the entry. It is when the viral DNA or RNA enters the host cell. Number 3 is the integration. Instead of immediately taking over the host, the viral DNA integrates into the host cell genome. Number 4 is the dormancy. It is when the viral DNA remains inactive and gets copied along with the host DNA when the cell divides. And the final step is the activation. It is when certain conditions such as stress, UV light radiation, or the immune suppression trigger the virus to exist the lysogenic phase. And now we will talk about retrovirus. It is when a group of viruses that uses RNA instead of DNA in their genetic material. And the first step is the attachment. It is when the retrovirus binds to a specific receptor on the host cell. Number 2 is the reverse transcription. It is when the virus releases its RNA genome into the cell, and when the reverse transcriptase converts the RNA into double-strand DNA. Number 3 is the integration. It is when the viral DNA enters the nucleus and it integrates into the host genome. Number 4 is the transcription and translation. It is when the host cell unknowingly transcribes viral DNA into RNA. And the last step is the assembly. Is used to make viral proteins and new copies of viral RNA. They are assembled inside the cell. And now we have talked briefly about viruses and bacteria. Thank you.